I just want to touch on um, two things. Number one, um, guys, make sure you're typing your name in the chat box. I put instructions in the assignment. It says to type your name in the chat box before the lecture starts. If we have any type of discussion during, you're supposed to um, participate in the chat through the chat box, and then you're supposed to type your name at the end of the live lecture. So if you don't do all of those things, then technically you're not following directions. And if I don't see your name where it should be, then I'm gonna assume that you were not here. I don't take roll. This is honor system. I expect you guys to put your name in the chat box. Uh, when we start, if you come in late, put your name in the chat box. And there are rules of how far or 15 minutes or more. If you're late, you need to write a summary. But even if you come in 15 minutes or more late, you still need to type your name in the chat box. That's a given because that's how I keep track of attendance. Second thing I want to talk to you guys about is with your discussions and any writing assignments that you all use. I know there are a lot of tools out there that you can use, namely AI, to help you write those different types of discussions and essays and paragraphs. But at the end of the day, your writing should be your own. So it should be your thoughts, your words on the subjects. Um, where there's no specific uh language that addresses AI or the use of AI, there are tools out there that can detect if what you wrote was generated by AI. And that falls under the school's plagiarism rule. So using AI is considered plagiarism. So, I get it. You get in the bind, you're busy. Sometimes it's hard to gather your thoughts and get things together. And I'm all for AI using it to give you a shell, but at the end of the day, if you're going to use it, you still need to put in your thoughts, your words, uh, maybe change some words, change some of the what it gives you so it makes it your own. Just don't put, copy and paste exactly what comes out of AI. Um, because you have to be able to think through the processes. You have to be able to think through um your discussion and your essays. There's a reason why we're asking you to write because we want your thoughts and words on the topics that we are talking about. So I just want everybody to keep that in mind. So we'll go ahead and get started. Any questions about that moving forward? Okay. We're gonna go ahead and get started. The information we going over tonight is not it's kind of dry. Um, so we're gonna get through it together. What I will tell you, is I did reference some chapters for you all to review out of the textbook. Those chapters are actually very, they give a lot more detail or explain things a little bit better, along with the videos, that from Red Tech Boot Camp. You know, lecture time is for us to review these topics, but we only have a small amount of time. Um, you know, this is not normal setting, so I really try to keep your lectures to 45 minutes. Most of the time they run an hour because, I know you guys are either getting off work or, getting ready, or you're at home trying to prepare for the rest of the night. So I try to package it and kind of hit the high points. Um, so we kind of prepare you for your test and just give you a general understanding. But at the end of the day, you got to go to the extra step. You got to read the chapters if they're chapters, review the Red Tech Boot Camp videos to help you kind of get a better understanding of the material. What we're gonna talk about tonight, it's more, this is stuff that we learned in biology way back in the day. We're gonna review it. I will just kind of give you guys some hints. Anything that's like bolded in the presentation, I would pay attention to, because you may see it again. So first tonight, we're gonna talk about the cell and the structures of the cell. So this is something we learned probably in high school, about what is in a cell, what makes up a cell. We're not gonna dive too deep into this, because it should just be a review. So our cells convert energy for vital functions, such as conduction of nerve impulses, contraction of our muscles, supporting various organisms, and then transporting bodily fluids. The operation of our cell is food for energy. They give the cells energy. The oxygen to break down that food, and then water to transport those inorganic substances. And we'll talk about some inorganic substances later. So if we work in radiation, if our cells are exposed to radiation, damage could happen. That's why we are so adamant about protecting ourselves against radiation protection, because we want to minimize cell damage at the lowest levels. We know that there's a possibility of there being damage. We can't really pinpoint it and we can't really be sure, but we know that just from reading about even some of the things that you guys did last week, an assignment coming up about the radium girls, and some of the watches and all that, the radium watches. We know that exposure to radiation could not only be dangerous to us, but also to our loved ones down the line, because it can essentially affect our damage our DNA, and that can be passed on to our offspring. So we know that if our cells are exposed to radiation, they may behave abnormally. An abnormally behaving cell is a cancer cell, can be an example. They can be a cancer cell, or the cells can die, which can cause damage as well. So chemical composition of the cell, protoplasm. This is the chemical building material for all living things. Uh, the protoplasm of the cell is made up of cytoplasm, nucleus, and other organelles. They're responsible for metabolism, which turns proteins into energy, reception of processing food and oxygen, and elimination of waste products of the cell. With protoplasm, it includes small and large molecules called macromolecules, and these molecules are broken down into organic and inorganic compounds. So your four primary components of macromolecules are carbon, hydrogen, oxygen, and nitrogen. So the organic compounds that make up our cells, we have proteins, carbohydrates, lipids, and nucleic acids. Proteins make up about 15% of the cell. They are structural and enzymatic proteins. They are repair. They repair. Carbohydrate makes up about 1% of our cell. This is our starches and our sugars that fuel metabolism, give the cell energy, and is responsible for short-term energy storage. Our lipids makes up about 2% of the cells. This is the fats, fatty acids, wax, and oils. It protects, insulates, lubricates the body of the cell, and aids in cell digestion. And it is responsible for long-term energy storage. And then your nucleic acids. This makes up about 1% of the cell. This is your DNA. So it can either be double-stranded or single-stranded. This is our DNA is our genetic code. And then also our RNA. This is the single-stranded. And this is what interprets the genetic code. So like I just said, protein constitutes 15% of the cell. It's essential for growth and construction of new body tissue. It's essential for repairing injured or debilitated tissues, and it's composed of amino acids. So our carbohydrates are saccharides, 1% of the cell content. Um, it's made up of monosaccharides and disaccharides. So monosaccharides are going to be your glucose, which is responsible for it's going to be your primary source of cell energy, your fructose and your galactose, and your disaccharides, maltose, lactose, and sucrose. The purpose of carbohydrates is to provide fuel for cell metabolism, and this metabolism takes place in the liver and the muscle tissue. Lipids, like you said, constitutes 2% of the cell content. This is our fats, fatty acids, oil, wax. Uh, this is present in our body tissue, acts as reservoirs for long-term storage of energy, insulates and guard the body against environment, supports, protects organs such as your kidneys and your eyes, lubricates our joints, assists in digestive processes. And then lastly, your nucleic acids, which are about 1% of cell contents. This is where you have your two different types of nucleic acids, like we talked about, DNA, deoxyribonucleic acids. This is your master chemical, contains all the information cells need to function. So, like I said, it's your genetic code, and your RNA ribonucleic acid. This is how that genetic code is translated by your RNA. So now we move on to inorganic substances of the cell. So one of those substances is water. Um, Water is fundamental in sustaining life. It is responsible for the cell transportation system. It regulates acids, bases, and salts, and it helps to maintain body temperature. It is the most abundant inorganic compound, making up about 80 to 85% of the protoplasm. Then you have minerals or salts, which are electrolytes. They produce energy and aid in conduction of nerve impulses such as heart contractions. So right here, it breaks down some of the electrolytes, the important electrolytes in the body. So good to kind of be familiar with those. So here's just a breakdown of what we kind of talked about, our organic compounds, including carbon, hydrogen, and oxygen. When combined with phosphorus and sulfur, they comprise the essential major organic compounds, which are our proteins, carbohydrates, lipids, and nucleic acids. Our inorganic compounds do not contain carbon, which we see right here. On the organic side, it says our proteins contain most carbon, but our inorganic compounds they do not contain carbon. Most important inorganic compounds are going to be water and the mineral salts. So now we're going to touch on DNA and RNA. Your DNA is going to be the most important molecule in your body. If it's unrepaired, the damage that happens can lead to, number one, cell death and tissue reactions, which we call deterministic effects. And an example of a deterministic effect is the of cataracts. So that's why we protect our eyes when we're in areas that tend to have a higher exposure to radiation. Then also what can happen is cell mutation. And an example of cell mutation is a stochastic effect. And basically what that is, an example of that is cancer. Then we have our RNA, which is going to be how that DNA is translated. It is single strand. It contains uracil instead of thymine. Your DNA was made up of DNA is two long sugar phosphate double helix spirals. They are linked by pairs of nitrogenous organic bases, kind of like like you look at a ladder, those steps on the ladder are what those bases are. So you have four nitrogenous organic bases of your DNA. You have adenine, cytosine, guanine, and thymine. So your A bonds with your T, so adenine bonds with thymine. Your cytosine binds with guanine, so C to G, and then guanine binds to cytosine, like which is G to C, and then thymine bonds to adenine, T to A. So these double strand helixes uh basically is what in those rungs is what makes a chromosome. Our chromosomes are composed of protein and that genetic DNA material. We have 46 different chromosomes in the normal human body, with there being 23 pairs in each cell. Individual male and female reproductive cells and germ cells, uh, which are not paired, and instead, each of the germ cells has only 23 chromosomes, which form a full set of 46 chromosomes when a sperm fertilizes with an egg. So you have one cell that has 23, another cell has 23. When that sperm meets that egg and it fertilizes, it combines with it to total 46 chromosomes. DNA in every chromosome is divided into hundreds of segments called genes, and those genes contain information that direct cytoplasmic activity, control growth and development of the cell, and transmit various aspects of heredity, like your hair color, your blood type, and general body characteristics. Genes are going to be your basic unit of heredity. Next thing we're going to talk about, cell division. So there are two types of cell division that occurs. You have mitosis that occurs in somatic cells, and you have meiosis, which occurs in genetic cells. So with mitosis, um, like I said, this is cell division for your somatic cells. So what happens is your parent cells divides to form two daughter cells that are identical to the parent cells. And then what happens is basically once those cells divide, there is a equal distribution of all cellular material when they divide. So you have two cells divide into, you have one cell divides into two. And then when those two cells divide, there's an equal distribution of the cellular material in each of those daughter cells. So next, meiosis. So this is going to be cell division of your genetic cells. So like we talked about, genetic cell has 46 chromosomes. And then as it progresses through the stage of mitotic division, to produce two daughter cells with each of those 46 chromosomes. Then each of the daughter cells will divide, and they produce two granddaughter cells that contain 23 chromosomes. So this is basically this table breaks down what happens, how it kind of breaks down those genetic cells. So sperm cells are very radiosensitive. And the cell goes through, it's more radiosensitive at different phases. So at the G1 phase, this is pre-DNA synthesis phase. S phase is the synthesis phase. This is when the cell is going to be least radiosensitive. You have a G2 post-DNA synthesis phase. Cell is still radiosensitive through during this phase. And then you have your mitosis phase. Cell is still going to be radiosensitive through that phase as well. But your synthesis phase is going to be the least sensitive phase out of all of three of these phases. So when we say radiosensitivity, this basically is the degree to which a cell, tissue, organ, or organism is susceptible to the damaging effects of radiation. So radiosensitivity is going to depend on your age, because children are going to be 10 times more sensitive than adults. That's why we take a little bit more precaution with protecting children. The type of tissue, our lungs, breast, gonads, bone marrow, they're going to be some of our more sensitive tissues, with having exposure, large amounts of exposure or exposure over time. These different tissue types are more likely to develop cancer. Biological sex. So women are 35% more sensitive due to their tissue composition, their genetic factors, and hormones. So basically, biological women are going to be a little bit more radiosensitive than men. Then the type of radiation. You can have 10 milligram of alpha particles versus 10 milligram of X-ray particles. The type of radiation is going to depend on the effects as well. Your alpha particles are going to be denser, and they're going to tend to stay in one place, versus your X-ray particles are going to be a little bit lighter and they spread out. And then the rate of exposure. So what's going on with exposure? Is this just a one-time exposure, and how large is that exposure, or is this an exposure over time? So 10 milligram in one day is more damage than 10 milligram in 1 year. So if you have a 10 milligram exposure in one day, that potentially could cause more damage than a 10 milligram exposure in 1 year. So this is just showing that different damage that can happen to DNA with radiation exposure. You can have a base pair lesion damage, which basically you see the rungs or the kind of the stairs are connected, but for some reason something happens and it's exposed to radiation, and you get a break in one of those rungs. This is most common, and usually your cell can repair it. So this happens, usually our bodies can repair it and everything's fine. Next is a single strand break. So not only do we break the rung, but we also break the helix. So this is usually repaired without negative side effects either. Sometimes the body can repair it and everything be okay. The most damaging is gonna be a double strand break. So rungs are okay, but we break the strands. This is gonna be your least common type of DNA breakage. It is gonna be your most difficult to repair though. And a higher proportion of unrepaired or misrepaired genetic information is gonna lead to birth defects or some type of effect, DNA with genetic effect. So target theory, I want you guys to make a note of the target theory and understand what the target theory is. Basically, the target theory is an adverse radiation effect that are only observed when sensitive DNA target molecules in the cells are affected. So basically what they're saying is you're going to have an adverse effect because you have some very sensitive DNA target molecules. So if you look at our table or our illustration, it shows the molecule, and you see the radiation coming in. The radiation damage hits the molecule, but the body can potentially repair that without it being any adverse effects. Versus that radiation coming in and hitting that helix, causing damage and causing cell death, which the if the cell dies and it mutates, then that could potentially cause a a major genetic defect. So, direct versus indirect. So, how does that radiation get to the DNA? So, a direct action is an X-ray photon or a free electron interacts um directly with the DNA. This is very uncommon because the DNA is only 1% of the cell. So only a small percentage of DNA is damaged caused by direct action. Then you have indirect action. This is where the X-ray photon or free electron interacts with the water of the cell or the helix, which radiolysis, that's the term for that, but it is bolded. So y'all make sure you read up a little bit more on radiolysis. This is the most common cause of DNA damage because the cell is about 80% of the water. So when that electron or photon interacts with that water, because it's so much of that water, it is gonna be probably gonna cause some DNA damage. One way you can look at it in the video, the Red Tech Boot Camp video kind of breaks it down as well, is that you have direct and indirect damage. So you think of, you have a house, it's storming real bad, and you get a lightning strike. Where this is possible, that happens few and far in between. So basically that direct hit from the lightning strike causing damage to the house is going to be the example of direct. Um, Now indirect is going to be, you have that house, it's thundering and lightning, and that house has trees around it. So the lightning strikes the tree, which causes a fire, and the fire causes the house to catch on fire. So that's described as an indirect hit. Um, so if you are trying to grasp it, get your, wrap your brain around it, just think about direct, it takes a direct hit. Indirect is something else gets hit and causes the damage to the DNA. Also, law of Burgon and Thibodeau. Please read up on that as well. Need to be familiar with that. This law states that the most radiosensitive cells are going to be your immature cells, which are going to be your sperm cells at the early stages of life, sperm and egg ovum cells, your unspecified cells, those that haven't formed into a certain tissue type, and then your high reproductive cells, rate, which those cells which divide and multiply rapidly. So we have a table that shows us our lowest radiosensitive cells are going to be our brain cells. And then our higher radiosensitive cells are going to be our bone marrow cells. So this table gives us an idea of the radiosensitivity based on the different type of tissue or cell. Now there may be a little bit, if you look at this table, you may look it up online, or you may look in a different book. They may have some variations between these. But for the most part, it's telling us that our brain cells are going to be least radiosensitive versus our bone marrow, breast, lungs, colon, and stomach, it shows, are going to be your more highly radiosensitive. So they're going to be more likely to experience damaging effects if exposed to radiation. So oxygen enhancement ratio. This is the difference in biological harm from each radiation type. So we have different types of radiation: alpha, beta, gamma, X-ray. So this is gonna basically explain to us the difference of harm that could occur with each different type of radiation. So your OER for X-rays is about 3. So that means X-rays are 3 times more damaging in an oxygenated cell than a hypoxic cell. Um, so oxygen, so a aerobic cell is what we're referring to when we say oxygenated cell. So this cell is going to be more radiosensitive, because oxygen binds to its lesions. Cell death can occur, and this can happen with radiation therapy or fractionation. Then you have your hypoxic cell. This is going to be a cell that has little to no oxygen. It's going to be less radiosensitive. It is able to repair immediately. An example of this is you have a five gray dose destroying a aerobic cell, so an oxygenated cell, versus a 15 gray dose to destroy a hypoxic cell. So you can see that 5 gray can destroy your oxygenated cell versus 15 gray. It would take 15 gray to destroy your hypoxic cell. So we're just moving right along. Next thing we're going to talk about is radiobiology, linear energy transfer, LET, relative biological effectiveness, and RBE. So with radiobiology, this is a branch of science that deals with the action of ionizing radiation on biological tissues and living organisms by combining radiation physics and biology. So you do have a question about alpha, beta, gamma, and neutron particles, or how we would handle them. So this kind of gives you an example of the penetrating distances. You see alpha, it can be blocked by paper. Beta can be blocked. It can go through paper and be blocked by plastic. Gamma X-rays goes through paper, goes through plastic, and it can be blocked by lead. And that's the reason why we are shielded with lead. We have lead in our doors and our windows and areas that are around our radiation. And then you have your neutron. And neutrons can go through paper, it can go through plastic, and go through lead. The only thing that really can stop it is concrete. So just kind of talked about all these particles. Alpha particles, very radioactive. They have a positive 2 charge. They are very dense, but they're not very penetrating. So like I said, stop by paper. Your beta particles, they have a plus or minus 1 charge. They have no mass, so they're going to be a little bit lighter. They can penetrate biological matter, but they can be stopped by plastic, wood, or metal. Your gamma or X-rays, and that's what we work with, or we're gonna be working with X-rays mainly. It's not very reactive, no charge, but it can travel a great distance. But it can be stopped by concrete and lead. And then your neutron particles are going to be your most reactive. They'll be able to travel the farthest, and they are highly penetrating. And they can be stopped by very thick concrete or a large amount of water. So low versus high linear energy transfer. So some examples of low linear energy transfer are going to be your X-rays and your gamma rays. They're going to be classified as your lower linear energy transfer. So that means that they deposit energy at a longer distance. They're less concentrated. They cause less concentrated damage, and they lower potential for biological harm because they have differential absorption. Then your high LET, those particles, examples of those particles are going to be alpha, beta, and neutron particles. So they're going to be considered have a higher linear energy transfer. And the reason why is because they deposit energy at a shorter distance. They cause more concentrated damage. And then they have a higher potential for biological harm. So they can cause the higher LET particles can cause biological harm if they are implanted, ingested, injected, or inhaled. So one thing to just keep in mind, the higher the LET, the higher the RBE. So the higher the LET, the higher the linear energy transfer, the higher the relative biological effectiveness. So the higher the LET, the more chances it's gonna cause more damage. So I would just make note of this slide. So RBE, relative biological effectiveness, is the radiation's ability or effectiveness to cause biological harm. So RBE of any radiation is compared to X-rays having the energy of 250 keV. I will tell you diagnostic X-rays have a RBE of about 1. So RBE 5 means that if something is given an RBE of 5, it means that it's 5 times more likely to cause biological damage than a 250 keV particle. So many may result in the same absorbed dose. You may have the same absorbed dose, but still it can still penetrate and cause more damage because the damage is more concentrated. X-rays tend to spread out versus some of those like alpha particles and neutrons. They are more they tend to kind of sit. Once they hit, it's like they just kind of sit there because they're stopped once they kind of get there. So this is gonna potentially cause or put someone at risk of higher biological harm. So 3 terms just to be familiar with, LET, linear energy transfer, RBE, relative biological effectiveness, and then weighting factors, WR, which is radiation weighting factor. So your LET is going to be the average energy deposit per distance. Your RBE is going to be the ability of the effectiveness cause of biological harm. So depending on the energy, how effective will the harm be? And then the weighting factor is going to indicate the ability to cause biological harm. So once this energetic particle hits and potentially causes that biological harm, what type of tissue did it hit and what's the potential of it causing biological harm to that specific tissue. All right. So that's all I have for you guys tonight. Y'all have any questions about what we went over? Okay. I just want to just kind of emphasize to you all to read up on target theory, law of Burgon and Thibodeau, and talking about the cell division, the different types of cell division. So just keep that in mind. If you don't have any questions, then you all are free to go tonight. I hope you have a good rest of your evening. Thanks, everybody.